Hello, and thank you for watching my presentation. My name is Court Alexander, and today I'm excited to tell you about my research on airborne environmental DNA. Environmental DNA, or eDNA, includes any genetic material discarded by organisms into the environment. Think of the dead skin cells that you shed every day, or the DNA left behind at a crime scene. These are examples of eDNA, and they can serve as a useful research tool. Airborne eDNA is just a subset of eDNA that is carried by the air. The analysis of airborne eDNA is a relatively new field of study that provides a way to detect and monitor microbial, plant, and even potentially animal species non-invasively. This has potential impacts for invasive species management, endangered species protection, and much more. There's still a lot we don't know about the ecology of eDNA, which includes where it comes from, what it consists of, and what affects its occurrence and distribution, among other things. We think airborne eDNA from plants can be in the form of pollen, plant fragments, or even free-floating DNA, but it's not clear which of these is dominant. A better understanding of this ecology would allow for wider application of airborne eDNA analysis. With this study, we set out to describe the particle size distribution of airborne eDNA. Our samples were collected using Big Spring No. 8 dust traps at the TTU native rangeland from the 3 sites shown. This rangeland is an interesting site for plant research because somewhere around 80 to 100 different plant species have been documented just in this area. After wind-blown dust had accumulated in the traps, we rinsed the dust out with water and passed the rinse water through filters with pore sizes ranging from 10 microns down to 0.2 microns. DNA was extracted from the material caught by each filter using a D&EZ Plant Pro kit. We then used qubit fluorometric quantification to estimate the total amount of DNA in each sample. qPCR will be used to more accurately quantify the amount of DNA collected and to determine its sources. We will then be able to determine how much DNA came from all plants, blue grama, which is a wind-pollinated grass, genus Budalua, and honey mesquite, which is an insect-pollinated tree, Prosopis glandulosa. Our preliminary results show that the vast majority of the DNA was contained within particles greater than 10 microns in size. Smaller amounts of DNA were found at smaller particle sizes, as shown. This chart shows the cumulative proportion of eDNA that was caught by each filter. For instance, about 85% of the eDNA containing particles in our samples were bigger than 10 microns, and so were caught by the 10 micron filter, whereas about 90% were bigger than 5 microns and so were caught by filters with pore sizes of 5 microns or smaller. This data suggests that most of the eDNA in our samples is pollen, although DNA from particles smaller than 0.2 microns has not yet been extracted or analyzed, and the qPCR data will be needed to confirm this conclusion. qPCR will also allow us to determine the particle size distributions for plant eDNA, blue grama eDNA, and honey mesquite eDNA, not just total eDNA. In the future, the results of this study and others like it could be used to determine the ideal filter sizes for species-targeted collection of airborne eDNA. More samples will be collected and analyzed this spring, and we may use larger filter pore sizes to get better resolution of particles bigger than 10 microns. I would like to thank Dr. Barnes and Mark Johnson for their guidance and support on this project, and also Pi Squared for my funding. You may contact me at my email address, which is shown if you have any questions. Thank you again for watching my presentation.